Hello, everybody. Today we're going to talk about a really interesting subject, the physiology of nerves and muscles. That is, how do nerves transmit messages? And then if the message is for a muscle to contract, for example, how does the muscle actually, at the cellular level, make that contraction? So we're going to cover very most basic introductory material relating to these systems. We're going to start by thinking about how information is going to come into an organism. And so it needs to have some kind of sensory receptor. And then we're going to look at some different kinds of receptors and use example organisms that are not just humans and mammals, but across the animal kingdom. So we're going to be able to have things that detect mechanical stimuli, so mechanoreceptors, things that can detect chemical stimuli, so chemoreceptors, and then receptors that can detect frequencies, sort of wavelengths of the electromagnetic spectrum, right? So receptors. Light is an example of a component of the electromagnetic spectrum. So eyes are an example of electromagnetic receptors. For chemoreceptors, taste and smell are examples, and mechanoreceptors include hearing, as well as just our ability to feel that we're touching something. We're going to look in general at what a nervous system consists of, typically a central portion where information is processed, and a peripheral portion, which is the nerves that transmit those messages to faraway parts of the body. We're going to zoom in on how an individual nerve cell or axon is able to transmit a message along its length in a series of steps that are called action potentials. We're going to see what happens if the message is to cause a muscle to contract. All right, so how does that message get to the muscle cells? What happens? And then how is ATP actually used in the muscle contraction cycle? Now, locomotion may be a result of a coordinated set of muscle contractions. And so we'll briefly look at different types of skeletons and how the muscles are fastened to them in order to accomplish a task like locomotion. Right, so when we think about information coming into an organism, and then that organism having to respond to that information. There may be information that comes in that you don't respond to, but let's suppose there is. You smell the fresh hot chocolate chip cookies coming out of the oven. You hear the clanging of the pan on the counter, and you get up off the sofa and you walk into the kitchen because your housemate has gotten the fresh hot chocolate chip cookies out of the oven and you want to eat one. All right, so we have sensors, or places where a stimulus is going to be detected. So in general, we're going to call the thing that happens that our sensor is able to detect a stimulus. And in general, a stimulus is going to be some kind of energy that can be detected by any one of these varieties of receptors. So a receptor or a sensor, a sensory organ or structure. Now, what has to happen is that the physical or mechanical or electromechanical information has to be turned into a message that can be passed along the nervous system. So it sort of has to be turned from the kind of information that exists in the world around us into a very specific kind of information that is going to be passed along the nervous system. So we typically call that thing a nerve that it's passed along. And so we're not just going to have sensory input here. We're going to have transmission. So how does that message get from your ear to your brain? it is transmitted along a nerve. How does the message get turned into a message that can get transmitted down a nerve? Right, that process is called transduction. Right, or we say, what's a D? Or we say the message has been transduced, right? Remember when we're talking about turning the message that's in DNA into a protein? Well, the process of translation is sort of like this transduction step. We're turning the mRNA message into a protein message. That's the translation step. We're going from nucleic acid language to amino acid or protein language. At this step of transduction, it's some part of the structure of this sense receptor that has that stimulus, strike it, bump into it, whatever, that literally is going to cause an action potential to happen in a nerve cell that is adjacent. All right, so we have to basically turn whatever the thing you're trying to sense is, we have to turn it into an action potential. Then that action potential will get passed along to the brain. Right, how does the brain know what happened? Well, for the most part, our nervous system functions because it has physical connections between specific things and specific parts of the brain. So as information is coming from nerves into the brain, it isn't willy-nilly, it isn't coming from all places at once, right? The sensation that you've just touched something with your forefinger always comes from the same place. So basically, as you're growing up or maturing as a small child, your brain learns to recognize that sensation, right? So there is a specific nerve and a specific nerve fiber that it associates with that particular stimulus, right? So we have this hardwired connection between a particular sensor and the particular location in the brain whose job it is to register that that stimulus has occurred and then process that information. So in this diagram, it says integration, and that's going to happen in humans in the brain. Now, the brain is what we call our central nervous system. The central part of the nervous system job is to have information coming into it, to decide what needs to happen as a result, and then to send messages out to the rest of the body. So it's a central in that it's not in the middle of the organism. Some organisms it is, but central as in where the messages come to and go out from. And depending on the organism, there may be a good deal of integration and processing. We would call that thinking about what needs to happen. In other organisms, we simply have a set of stereotyped responses, more like reflexes that the organism is going to do. Right, so this integration, another way to think about what's happening in the central nervous system is perception. Right, when we perceive something, it's almost like we consciously register that we've noticed it. We're also interpreting and processing that information. So in the human body, there is almost no response to any kind of stimulus that doesn't involve some kind of information processing in our brain. All right, and that is called perception. So we hear a noise. The only reason we know what that noise is because we have memories of it, right? And that our brain is reminding us of those things. So there's all kinds of interesting examples in psychology class you might have seen of where we can modify how our brains are perceiving information and trick it. And also how those things can happen because of injury or because of inborn differences in how people's brains function. All right, so once we've perceived or we've decided what to do with this information, we want to have a response, right? And the response is also going to involve transmission. All right, so a different nerve is going to take the message from the brain to the thing that needs to respond, right? And these nerves that are not part of our central nervous system, basically all the nerves that carry information to the brain and all the nerves that carry information away from the brain, those are peripheral nerves, right? And the effector is the thing that's going to make something happen. Now, in this example, it's a motor output. Our decision is that we need to run away from the stimulus that we have received or run towards the chocolate chip cookies. And so we're going to do a physical body motion as a response. All right. We could also have a response be the release of hormones, right? We could have a response that is to think, all right? We could have a response to do nothing. All right, so we are also going to have transmission happening. Now, when we think about how this works, the whole nervous system has dedicated nerves that are responsible for carrying information from the sensors to the brain. It has different nerves that are responsible for taking information from the brain to the place it needs to go. So one important thing to keep in mind is that each single nerve cell or nerve bundle is going to be carrying information only in one way. It has a special job. And that at the point where the stimulus is received, that message has to be transduced, that is turned into an action potential. So we'll see when we start looking at the different kinds of sense receptors that where that's going to happen is at the cell membrane. When we open or close ion channels at the cell membrane, we allow ions to flow into or out of the nerve cell. That's going to be the way that the nerve knows it needs to do something, right? So resting nervous system or resting nerve cells, neurons, are going to maintain a certain gradient in ion concentrations inside versus outside the cell. So when our stimulus opens ion channels, it's going to cause a flow from one side of the membrane to the other. And that is going to be a signal. That's the message, right? Now that is going to keep propagating down the nerve by this process called transmission. And we'll see how that works in a little while. All right. So as we are looking at how the nervous system works, we want to look at what are these things we're calling nerves. All right, a nerve is made up of a series of linked together nerve cells or neurons. And the neuron has a characteristic shape. And so it is a neuron. All right, notice these red arrows on the diagram. The dendrites, those are the part of the nerve cell, dendrites, that are going to be receiving information from upstream neurons. So information in each neuron is going to flow in one direction. It has a central cell body, kind of a fat round part of the cell where the nucleus is. And then it has typically many dendrites and then a single axon. Now the axon is the way the message is going to get sent to the next neuron, right? Neurons can be quite long, but when we want to get information from one end of the body to the other, we're going to have to string a series of neurons together. And so the end of the axon, called the axon terminal, is going to have cell membrane around it. And in this little side box, it's showing the cell membrane, the end of the axon. It's showing it kind of blunt at the end. And it's going to be close to, but not actually touching, the next neuron in this pathway. In order for the message to get from one neuron to the next, right? So the end of the axon of one neuron to the dendrites or to the cell body of a downstream neuron, we actually have to convert this message that's passing down across the axon, which we'll see later is electrochemical. We actually have to turn it into a completely chemical message, right? So we have electricity, it can jump across a little gap, right? But this gap, this space in between these two neurons is called a synapse. And the synapse is too big for that electrical message to go across. So we actually have to release a molecule from the end of one neuron. And those molecules are going to go by diffusion across this little narrow tiny space. They'll get received by receptors on the other neuron, and they will cause transduction of that message to happen again, right? So we have to turn the message from an electrochemical message into a chemical message and back into an electrochemical message again, right? So these neurotransmitter molecules are released into the synapse, and then they're pulled back in again, right? And they're stored, and then they're released again, and then they're pulled back up again. So they're reused many, many times. Now, neurotransmitters are kind of a very special kind of hormone molecule that are not moving very far, right? And some words that we associate with neurotransmitters are molecules like acetylcholine, glutamate, glycine, things that we talk about as endorphins, dopamine, serotonin, a molecule called GABA, capital G-A-B-A, and a variety of other molecules that are going to be neurotransmitters. So there's specific ones, different kinds of neurons have them in different locations. They might release different ones to send different kinds of messages. It's very complicated and interesting. All right, so when we have passed the nerve message on to the next neuron, same thing's going to happen. It's going to pass it down its axon. And when we call something a nerve, or we see a nerve in the body of an organism, what we're seeing is actually a bundle of these axons. Right. So when we are having a detection of a nerve cell, sorry, I'm moving my Zoom stuff. The way that we're going to be able to detect a stimulus happens in one of two ways. Either the stimulus is going to somehow touch, right, the actual neuron. And so we have a very direct communication between the outside world and your nervous system. Oops, go back. So we have direct stimulus when the actual receptor cell is a neuron itself, right? So we have these mechanoreceptors in our fingers. There's actually a neuron, and when we press hard onto a location on our finger, what happens is that the little tip of the neuron actually gets bent because it's being pressed on, and that opens up ion channels and triggers this nerve that something has touched it, right? We may have other kinds of epithelial cells that are the sense receptor cells, right? We don't always want to have a neuron, the tips of a neuron hanging out in whatever space of the body. So in your nose and inside your mouth, where we have sense receptors for taste and smell. Those are epithelial cells that are on the surface of the inside of our nose and mouth. And we want to have a cell membrane that is actually at the surface of the body in those locations. So the proteins that are on the surface of that epithelial cell are the place where those scent molecules, whatever, that chocolate molecule that's floating around in the air that's going to go into your nose, it actually has to land on one of those cells and bind to a protein. And that protein is going to trigger an action potential. right? So when this receptor protein is touched, or the chocolate molecule binds to it, it sets off a process inside the cell, which you'll talk about in the cell biology class, the steps that happen, that on the backside of that epithelial cell is going to cause an action potential to be initiated, right? So basically a neurotransmitter ends up getting released. Same thing happens when we have a synapse between two nerve cells. We get a neurotransmitter release from the back of the epithelial cell. It doesn't have to be nerve to nerve, right? We're going to get a neurotransmitter that's going to trigger the depolarization of the downstream cell membrane and trigger that action potential and have it be passed on, right? So each of the cells lining your nose and your mouth may be capable of recognizing a wide variety of different molecules, right? Your brain has to process that information. You have to learn that certain combinations of molecules represent certain things, right? So when we have direct stimulus to a neuron, the opposite of that, or the other thing that can happen here when we have an epithelial cell getting in the way, getting in between, is indirect stimulus. All right. So there's a wide variety of ways that the sense receptor can detect different kinds of information. So we're going to look at some examples. We're going to start with some of the kinds of receptors that we have in our skin. We have some kinds of neurons that whose ends or whose dendrites just terminate close to under the surface of the skin. And the sensation we receive when those are touched or triggered is pain, right? So those are very close to the surface of the skin. There's other ones that are deeper. We have these mechanoreceptors that are going to be sensitive to gentle touch versus firm or strong pressure that's applied. We have these neurons that are wrapped around all the little hairs. So if you imagine the hairs on your arm or the hairs on your head, when those are bumped, right, when the hair is brushed, it pushes the hair to the side, and these neurons that are wrapped around the base of that hair, they get bent, right? So that bending of those physical structures in the neuron is going to open ion channels. So like they're physically bent, right? And those are going to open. These pressure ones, again, they're also physically bent. The heat and the cold receptors are really interesting. So remember how proteins, when we're thinking about enzymes and how they work, that we know that if we change the temperature, it can change the way the protein folds. So if we get our enzymes too hot, they're going to unfold, they'll denature. So other proteins will simply change shape. They won't completely denature, but they'll change shape when their temperature changes. And that is the ion channel proteins that we find in these cold and heat receptors, that there's a series of different proteins that have slightly different temperature ranges at which they change their shape, right? So we can tell if something's cold versus hot because of the sort of the relative level of change that is happening in these receptors. So interestingly, the ones that are in our mouth, the heat and the cold receptors, the reason certain foods taste hot and spicy or cold, like minty, is that they happen to also bind to these receptors. So when we say that a chili tastes hot, it's partly because it is actually triggering the receptors that normally would be telling us that something is physically hot in temperature. So there's this overlap in how those receptors end up getting, how they get used. All right, so let's see. I think I talked about this. All right, we're going to start by talking about mechanoreceptors. Mechanoreceptors are capable of detecting mechanical stimuli. You can think of these as a physical movement of a structure, right? So the hair movement that we talked about before is an example of a mechanoreceptor, and so are the pressure receptors. And we're going to look at a couple of examples of different kinds of mechanoreceptors in animals. And other kinds of mechanoreceptors may be able to detect pressure or force, touch, stretch, motion, or sound. All right, so that means our ears or other kinds of sense organs that we might see in other organisms, right? In insects and amphibians, we have structures that function more like little drums that are called a tympanum, right? So a tympanum is an ear for an amphibian or an insect. And we have ears that we have this sort of outside part of, but the inner ear, the part that's way down inside, is the part where we actually hear the sound. So those are also mechanoreceptors. Our inner ear also has parts of it that tell us whether we're oriented right side up or not, and when we're changing direction. Right, so we have these little semicircular canals in the ear, and they are oriented in a mutually perpendicular way. And there's these little stones inside them. And as your head moves, these little calcified bits move around, and then there's little cilia inside, and your brain can interpret the information of the movement to orient you in that XYZ three-dimensional space. So similar organs exist in lots of kinds of animals and even in plants that work in a similar way. And here's a really, really simple version of it. So this body organ is called a statocyst, right? Notice that it has this statolith, and the stato part is related to the function of this body organ. Now, it's a very small body organ. It's not that many cells. It's a statocyst, and it's found in crustaceans like a crayfish or a lobster. And it's a sphere that has cilia pointing in the inside of it. And then it has these little calcium spheres. So when the organism is sitting still, those little balls or those little grains fall to the bottom, and they're deflecting the cilia, the sensory cilia that are on that side. So whatever side those neurons are getting triggered or activated on, because they're detecting this deflection, the brain interprets that as being that's the side that's down, right? So crayfish learned this when they're little tiny crayfish to be right, how to be right side up, right? So if they get swooshed away by a rapid river and tumbled around, then they can figure out eventually whether they're right side up or upside down, and then turn themselves over. Right, the fact that this, what this organ does was investigated by scientists who replaced these little balls with magnetic filings somehow. It's a tricky procedure, probably. And then they put a magnet on the top of the head of the crayfish, and then the little balls went up, and then the crayfish turned themselves over because they thought they were upside down. right? So possibly cruel, but also interesting way to find out exactly what this little organ does. All right. So that's basically the way that those semicircular canals in the human ear work. You can occasionally get a situation where some of those little stones have fallen out of place, or they've gotten stuck in a part of your that part of your ear. And there's a maneuver you can do to a patient and have them tilt their head back and turn to the side and whatever, in order to get those little stones back into the right place. And until you do, the patient is experiencing severe vertigo. They get really dizzy. Not pleasant, not life-threatening, but not pleasant. All right, here's another nice example of a mechanoreceptor. When we are following a school of fish around, or we're trying to scoop a fish out of our aquarium with a net, it seems to be able to avoid us so easily. Wherever direction you go, it swims away from it. So the way that they can tell is by a mechanosensory structure called the lateral line. And the lateral line, we can see it down the side of the fish, right? But when we zoom in on it, it isn't actually a line down the surface like a ribbon stuck on the outside. It's actually a series of structures that are connected together underneath the body surface. So imagine that you've turned your trout on its side and you've sliced down into it, and now you're looking at a section that is perpendicular to the body surface. So what we're seeing is this little open canal or tube that goes just underneath the skin of the body of the animal, and it has these little openings that are continuous with the outside water environment. Inside the canals are these little cupulas, right? So they're like a little dome, and when you press the dome to the side, it bends. These sensory hairs or cilia that are inside of it, and as it is bent, right, it deflects or bends. These sensory hairs, their cell membranes flex, ion channels open, action potential is transmitted, right? So if you're swimming towards a fish, that creates a pressure on the side of the fish because water is incompressible. So if you go towards the fish, you're pushing water ahead of you, it can detect that water is getting pushed towards it and it will swim away from you. Right, so these little cupulas, I don't know if you've ever had a computer with a one of those little pencil eraser nubs, or even if you think about a joystick on a game controller of any kind, this is basically how they work as well. If you bend it to one side, it makes an electrical connection on one side of that controller that sends a message to the game console that you want to move your whatever it is, your player, your wheel, your ball, your sword, whatever it is, you want to move it that direction. All right, so those are examples, again, along with hearing, of mechanoreceptors. Now we're going to turn our attention to chemoreceptors. So our example of chemoreception is the sense of smell, or olfaction, right? Our olfactory sense is here on the slide, right? Our sense of smell. Now, in most organisms, especially aquatic marine organisms, insects, other kinds of invertebrates, snails, they don't have a little pocket of air that's going to go inside their face. They're actually going to be detecting chemicals in the environment on their surface, right? So it's more like taste for them than smell. So a molecule, these molecules are going to be things that we can perceive as smells. They're called odorant molecules, right? So if you're, sniffing that pot of soup that's bubbling on the stove, or that chocolate chip cookie I was talking about earlier, the reason you can smell it is that molecules from it have volatilized. That is, they have gone from being in a solid state to being in a gas state, right? And so they're floating around in the air. And when we have a molecule or we have a liquid that's volatile, often that means it's flammable, but it also means that it's going to have a strong smell because it tends to go into the air very easily. All right, so that's fine when you're thinking about, oh, I'm tasting food that I'm putting in my mouth, I'm smelling smells that I like to smell, right? But same thing with dog poop. The reason you smell dog poop is because some of the molecules from the poop have floated up off the top of it and gone into your nose. Yicky. All right, so what we're seeing in this picture is we have two different kinds of odorant molecules. We have the butter smell and we have the chocolate smell from our cookies. So these are different molecules. They have a different shape. And the receptor proteins that are on the surface of these cells are going to be distributed around the inside of the nose or on the tongue. And those odorant molecules have to bind to, just like enzyme and substrate, they have to bind to a receptor protein that matches to them, right? So there are some flavors or tastes or smells that humans cannot taste or smell at all. We do not have any kind of a receptor molecule that fits to them. Humans have about 1,000 different genes. Other species may have more like 10,000 different genes for making these receptors. So more complex smells, we're going to have some combination of these different molecules that are going to all be triggering our action potentials in different neurons that we recognize as different tastes, and our brain is perceiving that. Right? Our brain is putting together that combination of smells and telling us it's that particular thing. So when we are talking about chemoreceptors in humans, we have these, basically the only ones we have for sense reception are our smell and our taste inside of our bodies. We also have chemoreceptors, and they function in our osmoregulatory system. They detect the presence or absence or even the concentration of different molecules circulating in the bloodstream. We need to know how much sodium ion concentration we have so that we can regulate it. Animals like insects are going to have a variety of chemoreceptors all over their body surface. They're going to have them on their feet, they're going to have them on their face, and then on their antennae. Right? So those antennae that arthropods have are going to be mainly chemosensory receptors. And when they're waving them around, they're basically sniffing. They're going to see what's going on in the air around them. Other organisms like fish may have these receptors on little whiskers they have. And so they may be able to kind of wiggle those around in the mud under their face and detect if there's a prey item there. All right, so that's chemoreception, basically the presence or absence of particular molecules or the concentration of those molecules. All right, our next set of receptors are those that can detect something that has to do with the electromagnetic spectrum. Right. So the electromagnetic spectrum includes electricity, magnetism, light that the human eye considers visible, light that the human eye doesn't consider visible. So off the end of our visual spectrum into the ultraviolet or into the infrared. Right. So we have organisms that can detect all of these different types of radiation. The reason or the way that that happens is that electromagnetic spectrum, it has certain wavelengths of energy that are represented along its spectrum. And that there is a protein in these body structures that when wavelength of energy interacts with the protein, it changes the shape of the protein, right? So just like the photosynthetic pigments in plants are going to interact with certain wavelengths of light in order to put an electron into a higher state of energy, right? We can also have proteins that interact with a certain wavelength of energy. And instead of putting that energy into an electron, they cause a ion channel to open in a nerve cell, right? So plants actually have the ability to detect light and dark in a way that allows them to bend towards the light or grow towards the light, which helps them do photosynthesis. So not only are they capturing energy from those wavelengths of light, but they do have some ways of having sensory reception, detecting the light and changing their behavior, right? It's obvious in animals that we do this. If you're stumbling around in the morning, you may gravitate towards light so that you can see where you're going. Animals have a very strong response to go towards or away from light. Many small animals that need to stay moist will have a strong behavioral aversion to light. They'll have a negative response to it. They'll crawl away from it. So they need to be able to detect it. And so we have neurons that are going to be in these visual receptor structures. All right, we think of them as being an eye. And this word ocellus, it just means a simple eye. And simple meaning that it can just detect light versus dark. All right, it can't form an image like our eyes do or cephalopod eyes do, but it can tell the difference between light and dark. And it turns out that once you can do that, you can survive if you are a flatworm. All right, so light is going to strike the neurons in this light detecting body organ. There are these molecules called visual pigments in there. Those are going to be those molecules that when the light strikes them, they change their shape. They're going to initiate an action potential in a neuron, and that message gets passed along to the brain. Now on these flatworms, they have these cute little cartoon eyes. And the reason there's two different colors on them is that the light is screened from one side. So the way this eye structure evolved to make it better and more useful is so that the organism could tell what direction the light is coming from. Not just that it's up there somewhere, right? But that it's coming from the left or from the right. So by shading one side of each of these eye-like structures, it allows the brain to perceive or interpret the information. Oh, I'm only seeing this light from my left eye, right? That means that it's dark to my right and I'm going to go that way. All right. So all we need to have in order to have an eye or eye-like structure that can detect light versus dark is to have these visual pigments, these molecules that change their shape when they are struck by light or electromagnetic radiation of a certain wavelength, and then they can cause an action potential because of that. All right, so we have more sophisticated kinds of eyes across the animal kingdom. One phylum that is very famous for its eyes is the phylum of the arthropods. And they have an imaging eye, right? If a fly is looking at you, it does see a picture, right? So it can see more clearly than if we are just detecting light or dark. It can have an image. Now, it's not focusing in the way that our eye is. When you hold something up close or far away, that you can really make modifications to the shape of your lens with these tiny muscles in your eye, like when you're focusing a camera to be able to see it clearly. So this is a special kind of eye called a compound eye. Right? It's not a simple eye. A simple eye or ocellus can only detect light from dark. Compound eye is a special kind of image forming eye that the arthropods have. And it's called compound because it's made of multiple repeating units, right? Each of these repeating units is called an ommatidium, and ommatidium is down at the bottom of this slide, that word. So each of these single ones, it's like a little column or a sort of a straw. But it has a cornea, like our eye has a cornea cap on the top. It has a lens inside there. The lens focuses the light coming through the cornea onto the photoreceptor that's inside of it. Now, arthropods are going to have a bundle of these, right? Some simple arthropods may only have a handful of these ommatidia, but your large, active, and successful organisms may have hundreds to thousands of these repeating units. So each of those surfaces, each of the corneas, is one of these little facets that you see on the fly eye. It looks like it's made of all these little dots. Each of those dots is one of these ommatidia. Each of them forms a complete image. And so the image that the fly gets from its eyes is actually multiple tiled versions of the same image. That gives us a headache when we try and look at it. But the fly brain is used to perceiving information that way. And what it's really good at is comparing all of these different images to one another to see if something's moving. All right, so it's really good at detecting motion from this pattern of visual information. And if you've ever tried to swat a fly, you know it can see you from almost over its shoulder anywhere and can fly away from you. All right, the human eye and the octopus eye or cephalopod eye work almost exactly the same, but were evolved by different, evolved based on, separately based on our evolutionary history. So we have an imaging and focusing eye. So if you've done your physics on lenses, then you know that as light is passing through a lens, it changes the angle the light is going, and that we are able to have light coming from far away or from coming closer to the eye. Both get focused onto the back of the eye because of changes in the lens. So if the lens is thicker or the lens is thinner, it changes the angle that the light is passing through. And our photoreceptors are on the back of the eye in this region called the retina. So we want to have the light striking the back of the eye here. And so notice in the figures here that the lens is fatter on the left-hand side and it's skinnier on the right-hand side. And that changes the angle that the light is deflected as it's passing through the lens. The way we do this is using tiny muscles that are at the top and bottom holding the lens in place in our eye. The eye lens is slightly flexible, and so it sort of gets stretched in order to make these changes. So in a terrestrial environment, that works great. But when we're in an aquatic environment, the ability to deform or flex the lens is not so great, because if you're at a deeper pressure, then the eye lens is going to get deformed. And so what we see in marine and aquatic vertebrates is that instead of having a thin flexible lens that's more lens shaped like we normally think about it, their eye lens is a sphere, and they focus it by moving it to front and back rather than up and down. So that's an alternate model of being able to focus the eye. So if you are ever able to do a dissection of a fish or a shark or a squid, you can dissect the eye, and what you find inside of it is something that basically looks like a sort of a whitish milky marble. It's the lens, and it's a sphere. All right, so this eye, we find it in vertebrates, and cephalopods, And this is an imaging focusing on. Right, and we only have one lens, so it's like a single vision, single lens reflex camera. All right. So now we've looked at a variety of different sense organs. So what they're going to do is create an action potential that's going to go from this sense organ to the brain. And perception is going to happen in the brain. And so I like to take just a very, very short moment to pause and think about, when we look across all different kinds of animals, we think about interpreting information and decision making, and that some of those components that we think about as intelligence. And when we look at different kinds of animals, we see that larger brains typically are going to be associated with larger brain per body size, be associated with higher intelligence. But when we have organisms that come from widely different environments, have different kinds of body structures, have different kinds of social structures, we have this list of characteristics that we as humans think we consider marks of intelligence or the ability to do certain tasks. And we have, all these interesting psychology experiments where we try having monkeys and apes and parrots and spiders and octopus and dolphins try and do these different tasks and try and interpret if they are performing them to the same kind of cognitive function level or intelligence that we like to believe that we humans have. So this cartoon is funny to me because the dolphins are speaking Spanish and the scientists are not realizing it because they're not expecting the dolphins to. So whenever we're thinking about what intelligence is comprised of, our own way that we perceive information, the way that we experience the world, because we're bilaterally symmetrical and because we have hands and because we have certain characteristics as primates, is always going to be influencing those conversations. And so it's really fascinating to study these things. When we have tried to use different kinds of intelligence tests on humans, we always find that those are biased in some way, that we have really not as good of an ability to discern how people function cognitively, and even trying to put it on some kind of hierarchical scale maybe is the wrong direction to go in the first place, right? So we'll come back a little bit more to that when we talk about the human brain. All right, so when we look at all different kinds of animals, and remember we're talking about the nervous system, only animals have a nervous system. Plants don't have nerves. Now that doesn't mean they can't transmit information around their bodies, but they do not have nerves. They do not have nerve cells. Like we talked about with nerves or connected body structures that transmit electrochemical nerve sensations from one place in the body to the other, that there may be a central structure, a brain-like structure that's going to receive that information and process it and then disperse instructions out. And we do see that almost every phyla of animals does have their nervous system separated into these peripheral, or to the side, and central, in the middle, parts. Now exactly what that looks like depends a little bit different. But in all of these organisms, all these different phyla, from echinoderms to flatworms, chordates, mollusks, that we have some central structure and then these sort of long nerves or nerve cords. The exception is this animal to the top left, Hydra, a cnidarian. And this phylum is unique among animals because it has no central structure. There is no place that messages get sent to and then sent back from. It's basically every neuron in its body is equal. It doesn't have any hierarchy. And so a message that's detected in one place on the body kind of just gets propagated around the body to the other side, and then it stops. And so their nervous system it allows them to do a series, some kinds of stereotype movements, but they can't think in the way that we think about thinking, what thinking is. Right, so they don't have any ability to process information or make decisions. Right, so this is the only phylum that has this nerve network that has no central part. All right, so I wouldn't even go so far as to say they have a nervous system. They have a network of nerve cells that are connected around their body, but that's all it is. All right, so all these other organisms have some kind of central part to their nervous system. Now, whether we call it a brain or not, partly depends on how big it is relative to the rest of the body. So basically, a brain is a big lump of neurons. And we can also have these other things, which are smaller lumps of neurons, which we call something else, which is a ganglia or a ganglion, right? So a brain is going to be a large blob of neurons functioning in a central nervous system role. Now, ganglia are kind of mini brains. When we have an organism with a fairly simple central nervous system, we may not promote it to the level of a brain. We might say, well, it has a ganglia, right? So here are our chitin. We don't honor it with the term of a brain, right? And some people wouldn't call the brain of a leech a brain. They would say it was a ganglion, right? So it's just a formality or convention. So these ganglia function in processing information as well. So they're basically accessory mini brains. And wherever we have ganglia, we may actually not need to send a message all the way to the brain. So we may have information coming in from sense receptors. It hits the ganglia makes the decision, and it sends the information back. So that message doesn't end up going all the way to the brain, right? Think about circumstances where there isn't that much thinking that needs to be done, and we want to have that response happen really rapidly because your life is in danger, right? So those kinds of responses, reflex responses, jumping out of the way of a bus that's trying to run you down. We don't need to make a decision if that's a good idea or not. We need to jump out of the way ASAP, right? So those messages, those kinds of decisions can actually be decided by the ganglia, right? And in chordates, we actually have a whole series of ganglia that go, every single vertebra has a pair of ganglia associated with it. Right? So lots of decision making is happening in your body without your brain even being involved. We look at other organisms whose body is made up in a segmented way. We typically also find these segmental ganglia. So in our annelid worms, remember leech is an annelid. It has repeated body structures in every body segment, right? Our arthropod, our insect, is also a body that's built in repeated segments, right? So we see these segmental ganglia as well. Each body segment has this little mini brain. And what that tells us about the vertebrate body is that indeed, through our evolutionary history, our body was originally formed with segments, right? So we don't see it on the outside anymore, but imagine our main skeletal axis with our vertebrae and our ribs. These are repeating units, right? So the vertebrate or chordate body is actually considered a segmental or a segmented body plan. And that means that as our body is being built during early development, it's built as a series of repeating units. We kind of merge them together on the outside later, so it's harder to see. But this is these segmental ganglia are an example of this hidden history of having body segmentation in our phylum. All right, we're going to get into some of the nuts and bolts about exactly how an action potential happens now. All right. All right, so it's a little complicated. We're going to go through the steps a few times, and it's really helpful to have a good understanding of what a neuron at rest looks like. All right, so neurons are never completely at rest. They're actually, well, they're not busy passing on an action potential. They're busily doing work in order to stay ready to pass on that action potential. They need to actively do work, right, using ATP in order to make sure that there is a gradient or a difference in concentration between the outside of the cell and the inside of the cell. And they're going to use the ions of potassium and sodium in order to create and maintain these gradients. And there are a variety of proteins that are transmembrane proteins. That is, they go through the membrane all the way. And they may be channel proteins. So channel proteins are going to allow molecules or atoms just to go through them. They're basically an open tube. And then we also have membrane proteins that are pumps, right? So they are actively going to be grabbing onto certain ions and pushing them up their concentration gradient, right? So that's going to require ATPs. So we need energy to run a pump, but we don't need any energy to have ions move through a channel. They're going to do that by diffusion. All right. So all of our ions are going to want to go down their concentration gradient by diffusion as long as there's an open channel that's going to allow them to do that. All right. So they can and will be flowing down their concentration gradient until it's equalized. Right. So we'll see that there's a complicated way that the cell makes sure that even though things are in equilibrium, that there is a difference in ion concentration from one side of the membrane to the other. And that involves another actor, which is those chloride ions. All right, so make sure you are oriented to the fact that we're starting with a neuron here. Oops. And we're looking at a little slice of either a dendrite or an axon, it doesn't really matter. The yellow represents the cell membrane, and then the light yellow is the interior of the cell. So remember, the axon is a tube. And then outside the cell is shown in blue. So if we look at the concentrations, so those little bracket symbols represent concentrations. And we have a concentration of K+, which is potassium ion inside the cell, at a higher concentration than it is outside the cell. We have a sodium ion, Na+, that is higher outside the cell than it is inside the cell. We have a chloride ion concentration that is higher outside the cell. Right. And the little m, big M's are millimolar solutions. And then we have this mystery element, capital A. It stands for anions. Right? Remember, cations are positive, anions are negative. So that big A concentration is representing all the other anions or negative ions that are not chloride ions. And we have a high concentration of those inside the cell relative to outside of the cell. Right, so this is the resting state of an axon, and it's an active rest, right? It's busily maintaining this concentration gradient all the time. And so we have a relative concentration of ions that is negative to the inside of the cell and positive to the outside of the cell. Right, so if we count up all of the negatives and positives relative to one another, then we find that adds up to this balance. Right, so the way that this resting potential is maintained, right, so resting potential is going to be the state that we're talking about with an axon that's at rest. And this potential means that there's a difference in concentration from one side of the cell membrane to the other. All right, so this is going to be how resting potential is created and maintained. And then after an action potential happens, we need to go back to resting potential and get ready for the cell to do this again. All right, so we're going to look at this diagram on the right-hand side and notice that the sodiums are yellow and the potassiums are orange and the sodiums are circles and the potassiums are squares. Right, so when we just squint our eyes, we see that there are more of the yellow circles to the outside. That matches our concentration in our little panel to the left. Higher sodium ion concentration outside the cell, and then we have a higher potassium ion concentration inside the cell. How do we make that happen? Well, we have the sodium potassium pump. All right, the sodium potassium pump is going to be working all the time. Part of the reason you need energy during the entire day and night is that your nervous system is continuously using ATPs in order to maintain the resting potential of your entire nervous system, and so that you can be ready to send nerve messages anytime you need to. All right, so notice this sodium potassium pump. What's it doing? It's pumping every time it pumps 3 sodiums to the outside of the cell and 2 potassiums to the inside. All right. We also have these potassium channels and sodium channels. Look at the diagram and see if they occur in equal numbers. All right, this is on purpose. Okay, so you should notice the sodium channels, the lighter purple ones, there's few, and of the darker purple ones, there's many. All right, so what we have going on is that we're busily pumping sodiums to one side of the cell. Right now, there's a concentration gradient. There's more of them outside the cell. So they're going to flow by diffusion back, right? But there's not that many of these sodium channels. So they can't necessarily go as fast as they want to, right? If all of the channels were closed, they couldn't go to the other side of the membrane. And the rate at which they can flow is going to also be determined by how many of these channels are open at one time. So we do have more of the potassium channels. We have potassium being pumped from the outside to the inside, and then it is flowing down its concentration gradient. All right, so why are we doing all of this, and what is the end result? Why are we pumping these things to one side and then letting them flow down to the other side? Well, it turns out that we get to a dynamic equilibrium. And the reason for that is these negative ions. And so when we have a certain concentration of positive ions on one side of a membrane, and we have negative ions on the other side, they exert a little pull. So as long as we have negative ions on each side of the membrane, the flow of the positive ions is eventually going to stop when it's balanced out. All right, so we're going to look at each the sodium and the potassium separately and see how the math works on that. All right, so if we're just looking at the potassium and the chloride ions, we have inside, we can set this up inside of a beaker, and it works exactly like it does in the nerve cell. So if we put our potassium chloride concentration on one side, and we've got these ion channels, there's only a certain amount of flow because these chloride ions that are on the left-hand side are going to exert a pull on those potassium ions. So the potassium ions are not going to completely be able to go down their gradient because the chloride ions are going to hang on to them. So we're going to end up, even though the potassiums can go down those channels, that they don't, right? And we end up getting approximately minus 90 millivolts charge from one side of this membrane to the other. You can actually calculate that using this equation that's at the bottom that you may have learned or used in your chemistry class and perhaps hated, but here is an actual really, really important biological use for these chemoelectric potential gradients, right? So when we have a difference in charge, a difference in concentration from one side to the other, we're going to call that a potential, right? The reason it's a potential is that we could let those ions flow down to the other side, right? And that they represent stored energy, right? So just like we talk about potential energy, we've rolled a ball or we've carried a bucket of water to the top of the hill. If we let it roll down, then it would change that potential energy into kinetic energy, right? And so these potential gradients or differences in chemical concentration and charge from one side of the cell membrane to the other is a kind of stored energy. All right, so the same thing is happening to our sodium ion gradient. And it is going to result in a positive about 62 millivolts difference based on the concentrations from the inside and outside of the axon in this example. So there's slightly different chemical concentrations depending on the kind of nerve it is, on the place of the organism that it is, and what kind of organism it is. So these particular voltages are estimates. All right, so the overall voltage difference between inside and the outside of the cell is determined by these two different forces of the sodium and potassium ions wanting to flow down their gradient. I won't make you do any math problems on this. All right, so our actual living axon is going to have a couple different things that can happen to it. All right, so we imagine that there are different kinds of these ion channels. Those are the ones that are going to be present in every axon, and the pump is going to be pumping all the time. All right. So there's other kinds of ion channels that can, under various circumstances, also open and close. So when we get a stimulus, it can cause, depending on what the stimulus is and what the kind of neuron is and what ion channel proteins are present, it can either cause a further polarization or it can cause a depolarization, right? So when we have an axon that's polarized, right, the way we're going to define it is that it's negative, remember last couple slides, that it's going to be negative on the inside relative to the outside. Right, and so the membrane potential, that's the difference in voltage from one side to the other. The resting potential in your average neuron is about minus 70 right here. Right, so if we look and see at something happening in the nervous system, we want to track what happens to it across time. So we can actually stick an electrode into the axon, especially if it's a large one. And depending on different organisms, we might be able to see very, very large axons, for example, in squid. Right, and we're going to be able to expose the organism to certain stimuli and see what happens. Well, some kinds of stimuli are going to cause a small hyperpolarization. You've got a polarization already. A hyperpolarization is going to make that polarization larger, right? So we're going to see that we have an even bigger polarization during a period of time, and then all of those ion channels that are normally open, right? So that's a hyperpolarization is going to cause certain kinds of ion channels to open. Ions are going to flow down them, down their concentration gradients. It's going to end up causing an even bigger polarization between the two sides of the membrane. But then those channels are going to close, the sodium potassium pump is going to pump, the ions are going to go down their concentration gradients through those always open channels, and it's going to return to normal, right? So the normal resting potential is something we're going to see in these graphs at the beginning of the time we're interested in looking at, and then it's going to return to that at the end, all right? So that is actively maintained by the pumping of the sodium potassium pump and those always open membrane channels. So no matter what happens to the axon, however it's disturbed from its normal state, it will go back to this resting potential. All right. So if we make the difference between the two sides bigger, that's a hyperpolarization. If we make the difference between the two sides smaller, that's a depolarization. All right. So on the second graph, we have these depolarizations, and they're going to cause the difference between the two sides of the cell membrane to get smaller. Now notice 0 is way up here, right? If we had 0 millivolts membrane potential, there is no gradient, there's no difference between the two sides, right? So we can have these small depolarizations that are essentially going to be stimuli that the organism doesn't need to respond to, right? So very small stimuli. If the stimulus is very small, then it just gets fixed, all right? We can hear a tiny little sound and decide we don't need to do anything, right? So very small stimulus that doesn't need to have any response to it. Right, so they're called depolarizations because they're going to be bringing the difference between those two sides of the membrane closer together. How that's going to happen is that we need to have a lot more ion channels open. That's going to allow both the sodium and one or the other of the sodium potassium to flow sort of much more vigorously from one side to the other. All right. So we're going to get these depolarizations if we have a stimulus that triggers more opening of sodium and or potassium channels. So if we have a weak stimulus, we just go back to normal. We have a strong depolarizing stimulus, right? We open a bunch of more ion channels. Then we end up getting a response, which is due to a positive feedback loop. So we have this complete firestorm of additional ion channels opening like crazy all over the place. Then they close. And then we have stuff flowing back the other direction, and then we return to normal. Right, but it turns out when we have this thing happen, that's called the action potential. It's a message that's big enough and loud enough that we can keep passing it along down the cell, right? So the way transmission is going to happen is that a stimulus that's strong enough to cause an action potential is going to be transduced into an action potential. And then that action potential gets passed or transmitted down the axon. All right, so the whole axon doesn't get depolarized at once. We end up getting a little tiny section of it depolarized. And then by diffusion, that depolarization spreads to the next little section. Right, so if we were able to monitor an axon and see what was happening to it, we had a stimulus hit it from this end, and we had a whole series of little electrodes in it. We'd see boink, a little action potential here, and then in the next little section of the axon, then we'd see boink, an action potential happening, and then boink, 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 boink. Right, so it's passed kind of like dominoes falling over one after another to the other end of the axon. So now we're going to look in more detail at exactly what is happening at each of these parts of the graph. Right, so it turns out the reason that something changes at each of these places where something changes on the graph is because there is a structure, right, that's doing a function. Right, so we're going to look at these different kinds of channels that are opening and closing, what happens to them, and then how the cell returns to normal. All right, notice on these graphs that time is on the x-axis, and that membrane potential in millivolts is on the y-axis. You have negative numbers towards the bottom. You go up through 0 and into the positive towards the top. All right, there's a lot on this slide, and we're going to spend a few minutes on it. All right, so notice our graph is in the middle of the diagram again. We have time on the x-axis, and then we have membrane potential on the y-axis. We start out in resting potential, and we have that panel on the lower left-hand side. So we have all the things that were going on in the resting potential state. We aren't showing them on these pictures because it would be too messy. We're just showing things that are different and new that are going to participate in the action potential. We have sodium channels and we have potassium channels, and these are different. These are not always open. They will open up if there is a change in voltage, right? So they act as if they have a gate that will open and close. And so these are called voltage gated channels. Right, so these voltage gated ion channels open if there is a change in voltage, and they have slightly different timing just based on the way that their structure is built. And the sodium channel also has this interesting little structure dangling down the bottom. It's like a little sink drain, right? A little sink drain stopper that you have on a chain by your sink so that you can close the drain if you want to, fill the basin up. It's called the inactivation loop. It works more like a stopper, and we'll come back to it later. So that's a structural difference that the sodium channel has from the potassium channel. All right, so when the neuron is at resting state, all those voltage gated sodium and potassium ion channels are closed. Notice that the ions are bouncing out of them, they can't go through. So there's a stimulus, and when we talk about this process of transduction, where we have some way that a stimulus is turned into an action potential, the most direct way that stimulus can get turned into an action potential is that it can open those it can open the voltage gated sodium ion channels. All right, so somehow this stimulus is going to open up some kind of ion channels that's going to allow ions to flow through, that's going to change the voltage. All right, so we start to have a small stimulus that opens some of these voltage gated ion channels, right? So notice we've got one of them that's open here, but not the others. The potassium ion channels are still closed. So we have a depolarization, right? We can have depolarizations that are not strong enough to get us to an action potential. But if we do, then, or we may have multiple stimuli that happen in a short period of time, right? If we get enough depolarization, right, to get to this threshold, right? The threshold level is a trigger after which there is a point of no return. Once we get enough of these ion channels open, we're going to open the rest of them. All right. So if we have a strong enough stimulus to cross the threshold, that means that enough of those voltage gated sodium ion channels have opened, that we're going to have a positive feedback loop that's going to open the rest of them. Right? So some of them open, it triggers the rest of them being opened. Right? That rest of them being opened is what's happening in this phase 3, right? The rising phase. So here we have a positive feedback loop. Once we hit the threshold, we've opened some of these sodium voltage gated ion channels. We hit the threshold, we open the rest of them. All right, so notice in our cartoon that we have both of these open. It's actually a lot more than that. Right, notice that the potassium voltage gated ion channels are still closed, right? They wait, right? And notice also that the inactivation loops are still dangling down there out of the way, right? We can't depolarize forever. We have to repolarize at some point. And so what happens at this point, right, at the change from this rising phase of the action potential, which is on the graph, the rising line, we go to the falling phase of the action potential, right? And it just is going down on the graph. Right, the thing that's different between this panel 3 and panel 4, right, a little while after all those voltage gated sodium ion channels open, they get closed with the inactivation loop, right? They get inactivated. What else happens at the same time? The potassium ion voltage gated channels open, right? So how do we get back to our resting potential? We've had this whole flood of sodiums going one way. Well, now we're going to stop that and we're going to let all the potassiums go the other way. Now we have all of our ions on the wrong side of the cell membrane now, but they're balanced out again, right? And so we get down to this, we return to having a polarized membrane. It's about as polarized as it was before, but it's polarized in the wrong way. And so then we have some cleanup to do. So in this step 5, we have some of the potassium channels are still open, the voltage gated sodium channels are closed, but the sodium and potassium pump is going like it always does. The sodium channels, not the voltage gated ones, but just the regular ones, those are letting the sodium flow back through. The potassium channels, not the voltage gated ones, the regular ones, those are letting the potassium flow back through. And we eventually get everything back onto the correct side again in the right proportions, and we end up at the resting potential. Right now, an important thing that happens is that until we get back to the resting potential, another action potential can't happen. We've got to get all our ions, our ducts back into a row before we can have another action potential, because they have to be poised, piled up on the correct side of the cell membrane to have those voltage gated ion channels open and have them flow down the correct way. So this little period of time when we're getting our ions sorted out again, right, we have this period of time when a second action potential can't happen. All right, it's called the refractory period. Right. We have refractory periods for other things. If you just ate a big meal, you can't eat another big meal again, right? You're in a food-related refractory period, right? You can't, you gotta wait till things are ready again. All right, so there's a lot going on here. What I want you to focus on, what's happening on the graph, why, what ions are flowing which direction, right? And what proteins, what are they doing? So which ones are doing what? And why does something change on the graph? I think that's the easiest way to organize thinking about how this works. So if you get a really good understanding of that resting stage, the resting potential and where all the ions are, then it makes understanding the rest of this you can go through things step by step. Once you understand the resting potential, everything that happens after it to make the action potential happen will make sense, right? That's what I had to do when I was learning this well enough to try and teach it to you all. I also have some animations and videos linked for you in the supplemental resources. If you didn't quite understand it from this lecture presentation, there is nothing wrong with you at all. This is one of the most complicated and difficult processes we have in the whole semester. It's really neat. It's really interesting. It's fun to learn how our nervous system works. But it's going to take a few times through it before you get your head around it. All right, so how does the way that this works, how does it make the action potential go from one little section of the neuron to the next, right? So we said that this is going to get passed along like dominoes falling over one after another. Okay, two things are important. One is that diffusion is happening inside the axon. There aren't little walls separating these parts of it. So that once those ion channels open and sodium starts to rush in, it's going to diffuse to the left and to the right. Okay, it's going to diffuse to the left and to the right. And so we don't need to open any more ion channels. We're actually going to get the sodium creeping down into the next section of the axon. And so we want to pass a nerve message from left to right here. So if this is the end, or the cell body here, and we have a depolarization happen, these sodium ions are going to keep drifting over into the next section. But we have enough of them in this region, they're going to start causing that action potential to happen, right? So the sodiums are all flowing in, then the potassiums are all flowing out. But what's going on in the next section? Now those sodiums have diffused down the axon far enough. Now they're causing a depolarization in the second little section of this axon. And so just like dominoes, these events are happening one after another, but kind of staggered, right? So we have the sodium ion channels open, the voltage gated ones open in this little section. Those sodiums diffuse to the next section of the axon, causing a depolarization, which causes an action potential, and so on. All right, now this sodium is going to diffuse to the left and to the right, but the sodium ion channels are closed with the inactivation loop in this little section here where the potassiums are flowing, right? So the sodium ion channels stay closed by the inactivation loop for a little while during that refractory period. And the reason that's important is that we couldn't move an action potential in one direction down our axon without having this refractory period, right? The action potential would just sort of keep going back and forth in a standing wave, right? The communication isn't going to happen if that happens, right? We have to force it to keep going in one direction, right? So while the first section is in its refractory period, the action potential that's happening in the second period can't trigger an action potential in the section that just had one yet, right? It's not ready yet, okay? So that's the mechanism that's going to cause the action potential to get passed down along the axon in one direction. All right, that's pretty neat. All right, so suppose we have gotten our message passed along, and this is an actual message that's going to a bundle of muscle fibers, and our response, we've transmitted this message and we want to make muscle fiber contract. Okay. Oh, sorry, skipped a step. Suppose we have multiple neurons and we need to pass this message from one neuron to the next in order to get it all the way from our brain to our left big toe. Okay, so we're going to have more than one axon. That means we have to have this message cross a synapse. We briefly talked about what happens at a synapse earlier. We're going to look at a little more detail. And the end of the axon, the presynaptic terminal, right? That just means that it's just before the synapse. We're going to have these vesicles or little membrane balls containing the neurotransmitter. They're hanging out there. They're ready to go. They're waiting. What's going to happen when the action potential gets to the synapse is that instead of there being more voltage-gated sodium channels and voltage-gated potassium channels, notice that instead we have a voltage-gated calcium channel, right? So our structures are going to be different. That means the function is going to be different, right? We have a different protein, a different structure. Now, when we open those, the voltage change, the action potential, it's going to trigger those calcium ion voltage gated channels to open. And the influx of calcium ion, the calcium ion is basically going to bind to those vesicles and tell them it's time to do exocytosis. It triggers the fusion of those vesicles to the cell membrane. All right, what does that do? It does exocytosis of the neurotransmitter. The neurotransmitter, shown by the red dots, is released into the synapse. It's a little space, it's a little interstitial fluid in there. It diffuses across that tiny space. On the downstream postsynaptic neuron, it's probably a dendrite at this end. All right, what do we have waiting for those neurotransmitters? We have another receptor protein, right? We want to bind to this neurotransmitter and have this message transduced, right? It has to, again, get switched from a chemical message into a depolarization, right? An action potential. How are we going to do that? The neurotransmitter binds to these ion channels. So instead of being voltage gated, that open when the voltage changes, they open when the neurotransmitter binds to them, right? Something that binds to something else can be called a ligand or a ligand, right? So we have these ligand-gated ion channels. That means their gates are opened when the neurotransmitter binds to them, right? So that's how the action potential gets passed from one neuron to the next across the synapse. The neurotransmitter is released and the neurotransmitter binds to these ion channels on the postsynaptic neuron cell membrane, opens up, lets the sodium potassium ions flow through down their concentration gradients and creates a depolarization. All right. Now we have an action potential in the next neuron and we can pass it along. All right, so now we're going to talk about muscle response. In order to understand how a muscle contracts, we have to look at the structure of the muscle. So when we think about what a muscle is, we look very top of the slide, and we think about our biceps muscle. It's a nice, big, bulging muscle belly that's going to be contracted when we move our hand towards our shoulder. That thing we call a muscle is actually made up of many, many subparts. Right. The biceps, for example, is going to be made up of several of these bundles of muscle fibers. And each of these bundles of muscle fibers is made up of muscle fibers. Each muscle fiber is a set. So like neuron is the name for a nerve cell, muscle fiber is the name for a muscle cell, right? So the fibers are the cells. They are multinucleate, right? So that means even though we have a single cell, we have many nuclei inside of that single cell. Within these cells, there are bundles of these protein fibers. So these protein fibers are called myofibrils, and they're organelles, basically. They are inside this muscle cell. So the cell membrane's around them, and then we have a whole pile of these myofibrils. So myofibrils are structures that are made out of actin and myosin proteins. And they're round in cross section. And what they're made of is actually, if you sort of zoom in on them, they're actually a couple different kinds of fibers that are stacked next to one another. Each of the, along its whole length though, they're sort of separated into sections, like train cars are separated into sections. There's a whole bunch of cars together, but each car has its own beginning and end, right? So we can see, if we looked at an electron microscope image, we can see the edges of these sections, and we call them Z lines. I don't know why. But these Z lines are kind of like a little disc. And when we look at how the overall muscle fiber contracts or shortens, we'll see that the Z lines get closer together, right? If we're doing a small muscle contraction, we don't have to contract our muscle all the way, then we'll see the Z lines get closer together, and they'll be getting closer together across the entire length of the myofibril. If we do a forceful contraction or a strong contraction, then the Z lines will get much closer together. They can't go all the way to touch one another, but they'll, right? So when we think about how a muscle does its job, it is by getting shorter. The process of the muscle getting shorter is called a contraction. And the way that happens is that each of these units of the muscle fiber, each are getting shorter or sort of squishing together. All right, so the Z lines are going to get close together. Each of these units, the functional unit of the myofibril from a Z line to the next Z line, like a car on a train, is called a sarcomere. We only have to look at what one sarcomere is doing to understand what's happening across the whole length of the muscle, because it's all happening exactly the same. All right, so in order for us to have this contraction happen, we have to have some kind of sliding or movement of one side against another. So it turns out there's two kinds of protein fibers that make up our sarcomere, and they're called M lines. There's an M line in the middle of the sarcomere, and the two kinds of filaments in the sarcomere are called thick filaments and thin filaments. Thank you, people who named these things, for the fun names. The thin filaments are made out of the protein actin, and the thick filaments are made out of the protein myosin or myosin. Now notice that the purple are the thick filaments, and that the myosin fibers are connected to the M line. Right, so you can think about the M line as kind of being anchored. It's going to stay in the same place. And then the Z lines are each going to move towards the middle. Right, so the M line's the middle, the Z lines are at the ends. The actin filaments, or the thin filaments, are shown in yellow. Each of these filaments is made up of many of these little proteins formed into these particular structures. Right, so I'm going to show you on this figure what's going to happen overall in muscle contraction. All right. So each sarcomere is going to have these thin filaments, or the myosins, fastened to the Z lines, and it's going to have these thick filaments connected to the M line. All right, what's going to happen in contraction is each of the Z lines is going to move towards the M line. So when we get a contracted sarcomere, M line's going to be in the same place, right? I may not be drawing it amazing, but the filaments are not like the lens of our eye. They aren't going to get fatter or skinnier. They're going to stay the same size, right? But what we want to see is that the Z lines have moved closer to the M line. And where we had these space in between the tips of the thin filaments and thick filaments, now there's more of the thick and thin filaments that are overlapping, right? So we started with them here, and in order for the contraction to happen, they've slid so that they're more overlapping, right? So relaxed at the top, contracted at the bottom. All right, and it still has the Z line, M line, and other Z line. All right, so we're going to look a little more detail at exactly what happens in that contraction, but I want to give you an overview before we get into the details. All right, so how does a muscle fiber know that it's time for it to do a contraction? All right, that's where we're going to start. This message has just come from the nervous system. How does it get there? All right, so here we have a synaptic terminal. So we have a neuron that is coming from the brain, and it's saying, contract this muscle right now. How does it do that? It releases the action potential gets to the terminal, which causes those calcium voltage gated ion channels to open to trigger the neurotransmitter release into the synapse. How does that happen? The vesicles fuse with the cell membrane, and then the neurotransmitter is released by exocytosis. The neurotransmitter in this case is acetylcholine. And what we have in the cell membrane, right, the surface membrane of the muscle cell, shown in that darker pinkish color, is we have, here's the neurotransmitter acetylcholine, we have ligand-gated ion channels, so they're acetylcholine-gated ion channels. So what happens is that the neurotransmitter causes a response. The acetylcholine-gated ion channels open. That causes a release of calcium from the structure, the squiggly green structure that's shown here. And this squiggly green structure that's releasing the calcium is called the sarcoplasmic reticulum. All right, we have endoplasmic reticulum that you're probably already familiar with, where we find the ribosomes, and we make the proteins that are coming with the messenger RNA is coming from the nucleus, right? There's endoplasmic reticulum in every cell. There's only sarcoplasmic reticulum in special cells, for example, muscle cells, right? So the message of the acetylcholine-gated ion channels opening is what triggers calcium ion release from the sarcoplasmic reticulum, right? The sarcoplasmic reticulum has been waiting there. It's sucked all the calcium up again from the last time that it released it, and it's ready for this muscle fiber to be triggered to contract, right? It turns out that while a muscle fiber is relaxed, or it doesn't need to do anything, it has the place on the muscle fiber, or sorry, the myosin fiber, the place on the myosin fiber that's sort of going to start the process, it has it blocked, right? So in order for the message for the myosin fiber to get ready to contract, we're going to unblock the binding sites on it. They're blocked by another filament called tropomyosin, right? So the tropomyosin is shown on here. It looks kind of like a string, and it's laying across these green ball shapes. Those green ball shapes are sort of dimples, and the actin filament wants to bind there, right? So the muscle fiber is going to contract if there's enough ATP, and if the actin filament can bind to the myosin filament. it can't bind to the myosin filament if this tropomyosin fiber is covering it, right? The troponin, there's a lot of T things here. The troponin is that little yellow structure. The troponin sits on top of the tropomyosin. And when calcium ion binds to it, it twists, right? It twists and it pushes the tropomyosin off of those actin binding sites, right? So when the calcium is released from the sarcoplasmic reticulum, it binds to the troponin. The troponin twists and it pushes the tropomyosin off of the actin binding sites. That allows the actin to bind, and with energy from ATPs, if they're present in the cell, then series of actions are going to happen that are going to pull that myosin fiber with respect to the actin fiber and will cause that contraction or shortening, right? So these actin fibers are the thin filaments, these little fingers that are sticking up, they're going to grab onto thick filaments and they're going to pull, right? And that is going to make the thin filaments move towards the middle of the sarcomere. And we'll look at a little more detail of how that happens in the next slide. The next slide after this one. So here we're zooming in on this process with the calcium ion is binding to the troponin, and the troponin twists, reveals the myosin binding sites, and it's going to, oh shoot, I think I just gave this explanation as if the actin were reaching up. Okay, take that back. The thin filament is the one that has the binding sites. The myosin head is the one that's reaching up. I apologize for that. My Zoom menu was sitting on top of that picture. All right, actin filament has the binding sites. All right. So here we are zoomed in. Thick filaments, or the myosin, have these little turkey drumstick things sticking up. Thin filament, or the actin filament, has these little dimples, right, which is where myosin heads are going to bind. There's ATP present in the cell. It's going to be sitting there ready, bound to these myosin heads. Now, when the myosin heads are in a low energy configuration, they're going to be kind of bent down. When the binding sites become available, the myosin head is going to break the bond between the last phosphate on the ATP, right, adenosine triple phosphate, and it's going to break the bond between what is left over, which is an adenosine diphosphate, or ADP, and a free phosphate. The ADP and the free phosphate are going to momentarily stay bound to the myosin head. The energy that was released from breaking that bond is going to push the shape of that myosin head from being folded over to sticking up, right? It's going to be sticking up. It's going to bump into that myosin binding site on the actin filament. And then it's going to release the ADP and the free phosphate. And in doing so, it's going to relax to its low energy configuration. All right, but it's already bound to the actin. All right, so this process puts energy in it. It causes it to reach up and stick to the actin filament. And then as it falls back down to its low energy configuration, that sort of falling movement actually drags the thin filament along with it. All right. So one of these little hooks pulling right, is not going to do anything interesting. But basically, they're all pulling at once, right, or one after another. And so there's always another myosin head that's ready to bind to the actin, right? So it's going to be doing this sort of, they're taking turns and they're continuing to pull the actin filament towards the center of the sarcomere. Right, so we have to get energy from an ATP molecule, and then that ADP and that free phosphate, it's going to be released. It's going to go back to the mitochondria and the cell. Glucose is going to get put through cellular respiration, and the phosphate is going to get smashed back onto the ADPs, released back into the muscle, the cytoplasm of the muscle cell, so that it can be ready to be used again next time. So after this process is complete, what happens to the muscle? When the myosin head can't bind anymore, the muscle is going to separate, you know, it doesn't get hold, it doesn't hold clenched, but it's going to relax, right? So as long as a muscle fiber is not being triggered to contract, it will be relaxed, right? And the way that a muscle fiber can uncontract, we don't have an opposite process where the heads are going to walk the other direction. We actually have all of our body muscles arranged in such a way that whenever there's one that contracts to do a motion, there has to be another one that does the opposite, so the body part can go back to where it needs to go again. Right. So when we see the way that the muscle fibers can only contract, they can't forcefully uncontract, go the other direction. So we have to have our body muscles arranged in a way that has these two different functions. So whether we have an organism with an internal skeleton like a human or other vertebrate shown on the left, we have a biceps muscle on the front of your arm, and its opposite muscle is the tricep on the back of the arm. All right, so if you flex your bicep, you contract that muscle. You have to relax your tricep in order to do that. It has to stretch and elongate. In order to straighten your arm again, the tricep muscle has to contract, right? Where we have organisms with an external skeleton, same thing is true. The physics of it's a little different. So here we have the leg of a grasshopper. It has to have opposing extensor and flexor muscles as well. They aren't arranged the same way. But one of them is going to sort of pull up on the end of this leg to make it go one direction. The other one is going to pull on the other side of that lower leg and make it go the opposite direction. Right, so really different physics and mechanics of an internal skeleton versus an external skeleton. For the size, the external skeleton organism is way, way stronger just because of lever arms and physics stuff. All right, so when we have a coordinated set of muscle contractions, we can get locomotion to happen. All right. We're just going to look at earthworm locomotion. If you want to read in the textbook about, jumping, hopping, flying, and swimming, go right ahead. This is highly exaggerated in this diagram, but it's a really interesting way of thinking about how animals do this complicated process of locomotion. So an earthworm only has two kinds of muscles. It has ones that wrap around it, they're called circular muscles, and then it has longitudinal muscles, the ones that go along it. So it doesn't have biceps and triceps and whatever. The middle of its body is full of water, right? So it has a hydrostatic skeleton. When water is squished, it has to go somewhere, okay? So the way that the earthworm is going to be able to do locomotion is that it's a coordinated set of alternating contractions of circular and longitudinal muscle. So when it's squishing in the middle of its body, that causes its head end to shoot out to the front, right? So it's going to relax its circular muscles in one place. It's going to contract them in another. The head end is going to get long and skinny, right? And then it's going to be able to anchor itself in the soil with these little bristles or setae that are sticking out. And its head end, now that it's poked it out ahead, it's going to anchor itself in the soil. It's going to contract its longitudinal muscles in order to pull up the last section of the body, right? So in the earthworm, this alternating contraction of muscles called peristalsis. Right, peristalsis is an alternating contraction of circular and longitudinal muscles. In the earthworm, it results in locomotion. Our intestine also has circular and longitudinal muscles. And the result of peristalsis happening in our intestine is that the lump of partially digested food matter that's inside of it continues to get pushed down the tube, all right? So that's really important in terms of organismal biology, functions that are happening in body organs inside our body. The same kinds of structures are building them and running them. In a whole other organism, they're a very similar structure, but the function is different. It's locomotion in an earthworm. All right. So we just touched the very, very tip of the iceberg in terms of understanding how a nervous system works. There's all kinds of neat stuff about how strong a stimulus needs to be, all different structures of eyes and all kinds of neat stuff about how the overall skeletal and muscle system works. If you're going to med school, you'll learn about it then. If you're going on to other things, hopefully this has been just enough to get you more excited about it.